We are ready.
Okay. Am I just start? Or?
No, I'll let Uri to introduce you.
Okay, sounds good.
So should we start?
Mm-hmm. Yeah, we shall start now.
Okay. So hello everyone, one more time. So thank you very much for attending to the EFSC Institutional Seminary. It's my pleasure to introduce you to the Professor Rio Yasuda, who is one of the scientific directors of the Max Planck Florida Institute in Neuroscience. And this is personal opinion. He also is a great scientist and is an amazing person. It's really great. So very short, briefly, he got his PhD in physics from Keio University Graduate School in Science of Technology in 1998 and Yokohama, Japan. From 2000 to 2005, he was postdoctoral fellow at Cold Spring Harbor Laboratory, where he built an imaging device to monitor protein interaction in living cells with high sensitivity and resolution. After that, from 2005 to 2012, he was an assistant professor on neurobiology department at Duke University Medical Center, where he developed several techniques to visualize signaling activity in single synapses. From 2009 to 2012, he also serves as an early career scientist at the Howard Hughes Medical Institute. Honestly, I could keep talking about the huge trajectory of the Professor Ryohei and a lot of awards that he has, but I think it's better to listen to him and the talk that he prepared for us. And thank you very much, Ryohei, to share this with us, and welcome.
Great, thank you very much, Uri, for introduction and inviting me. You hear me, right?
Yeah,
okay.
Perfectly.
Great.
Thank you.
Great. Okay, so I share my screen with you guys. Okay. Do you see the presentation? Yes. Okay.
Yeah, it looks great. Thank you.
Okay, great. So thank you very much again. So I'm going to talk about the neuronal intracellular signaling underlying synaptic plasticity. This picture is Max Planck Florida Institute. We are very close to Mexico, so hopefully we have some visitor from Mexico sometime. So let's talk about neuronal signaling and why we are interested. So probably you know that our brain action is governed by neurons that is firing. That basically provide us with a very fast and instant response or reaction to things happens. Right? So basically the neural activity of the neural circuit is playing very critical role in controlling our behavior in very fast time scale. And one of the important aspect of the neural activity in the brain is very fast, right? Using action potential, we can have a very quick response. And at the same time, our brain also can have much longer time scale, like to remember things, right? Or adapt to different behavior and kind of thing. So, so this is considered to be governed by intracellular signaling. And of course, there are a lot of different cells in the brain, but we are particularly interested in neuronal intracellular signaling. And we believe that this is important for long-term action of the brain. And that's why we want to understand what is how the intracellular signaling of neuron is regulated. Now, when we are talking about intracellular signaling in neurons, there is a particular challenge for neuroscience because neuron is a big cell. and this the cell has huge and very complicated processes, basically spanning more than 100 microns and sometimes millimeters for axons. Yeah, so or centimeters, of course, for peripheral neurons, and therefore the intracellular signaling in such enormous cells very specific regulation in terms of the space, right? So the localization of intracellular signalings are extremely important. Of course, in all cell biology it is important, but in neuron particularly have a very segregated signal transduction system in each cell, and yet the whole process in these small compartments or small to mid-scale compartments are not so well understood. So we are particularly interested in the signal transduction occurring in dendrites, and also a small structure called dendritic spines on the dendrite. Okay. For each pyramidal neurons, these excitatory neurons have the structure called dendritic spines, and they have typically 1,000 to 10,000 spines in each neuron. And this is a very tiny structure of 0.1 femtoliters. And this is actually receiving side of the synapses. So they are receiving the postsynaptic side, yeah, of the cell. So they receive the information through this compartment. And why we are interested in this structure? First, of course, it is important for synaptic transmission. But at the same time, we know that the structural change of these spines are correlated with animals' learning and memory. Right. So this is, for example, one of the model of learning memory. Basically, people found that spine changes its structure when animals learn, right? And that for example, newly formed spines or enlarged spines tends to be associated with learning of new experience, right? And some other spine that existed tends to be removed because each neuron have certain capacity of the memory. So there a lot of people think that these dendritic spines are the basic unit of learning and memory, yeah, and information storage, basically. And another aspect important for dendritic spine is that abnormal spine morphology is associated with mental disease. So it is well known that different diseases like mental retardation, schizophrenia, Alzheimer's disease, autism, almost all of them are associated with abnormal spine morphologies. It's interesting that sometimes the patients tends to have reduced number of spines, and sometimes too many spines. But it seems like actually appropriate regulation of dendritic spines are extremely important for our normal function of the brain. So it is kind of understandable. We need to understand what's going on, how the spine regulates its function and the structure. Now here comes our problem of signal transduction. Basically, there are enormous number of proteins that is important for synaptic plasticity, so-called synaptic plasticity, that is important for changing the synaptic weight. So these are the protein. Probably, if you are cell biologists, many of these proteins you might have seen, right? Like pKa or let's say Rho kinase, sorry, Pkc, Rho, and Ras. These are all kind of very well-known proteins. So it's a kind of simplified diagram of intracellular signaling, and it's very similar to actually almost all other cells. There are some neurospecific proteins, but actually the components are relatively similar to other cell types. But the difference is, of course, the neuron have the very tiny compartment right, like spines, and within 0.1 femtoliter, this they have this enormous circuit of signal transduction, and this basically plays a role in transmitting the activity of the spine, right? Usually calcium triggers signal transduction. And then somehow these components basically changes acting regulatory proteins and produces signal transduction that is important for acting cytoskeletal changes, right? So that kind of thing. So basically, we want to understand what's exactly going on in this tiny structure. So our lab has been working on 3 different projects, basically to develop tools, right, to understand this signal transduction. And actually, probably the tool is generally useful for almost any aspect of cell biology. Today, I'm going to talk about the monitoring activity of different proteins in cell. Right, intracellular signaling can be monitored with our technique. So our technique is based on 2-photon fluorescence lifetime imaging microscopy. So this is a microscope combining 2-photon microscope and fluorescence lifetime imaging microscopy. We started this project like 16 years ago. Basically, the idea is using fluorescence lifetime imaging, which can measure threat very sensitively and also with a very high precision, right? And this report of molecular interaction complexity. So fluorescence lifetime we can measure by exciting fluorophore with short laser pulse and measure the decay occurring in nanosecond time scale. Yeah. When the molecules bind to each other, they decay faster. When they are separated, they decay slower. And if in mixed population, we have this different decay component we can fit to measure the binding fraction. And importantly, we need to have sensors in the cell, like to report us the activity of the proteins. So in the past 20 years, we developed a lot of different sensors, and these provide us with very important information about signal transduction, which I will show you later what kind of information we get. But you can see actually what we developed, like kinases. Some of them are also really worked on. And small DTPase protein, transcription factor, and so on and so forth. It takes very long time to develop each sensor, but then it provides us with very important information about intracellular signaling. Another important aspect we use for this particular talk, is that we use single spine stimulation using glutamate uncaging to photon glutamate uncaging in brain slices. So basically we label 1 neuron in the slice, right? In this case with GFP and sometimes with a sensor. And we stimulate 1 spine. You can see that actually in response to glutamate, which is a neurotransmitter for this particular synapse, yeah. Basically we observe big structural change and enlargement of the spine. Okay, so again, we stimulate, and then you can see actually it becomes bigger. This volume change occurs very rapidly in response to glutamate uncaging. And then it decays over a few minutes, but it remembers the stimulation over a much longer time. So the size, like this is a volume change, right? So the size remains bigger, right, like 100%, it's twice bigger over more than 30, 40 minutes. Actually, we have measured 3 hours and still the size remains bigger. I think actually it's probably will be bigger for a very long time, maybe days. And actually the function of the synapse also changes, right, in response to this. So in the stimulated spine, we observe larger glutamate sensitivity, right? So when we glutamate uncage again, again on this spine, we observe much bigger response to the same amount of glutamate. suggesting that we have more glutamic receptors, or maybe maybe each receptor becomes more sensitive. Now, when we measure the glutamate sensitivity on the stimulus spine, we observe this kind of enlargement. But when we measure adjacent spines, we don't see any change, right? So this suggests that we are actually inducing plasticity only on the stimulated spine, not surrounding spine. It's a spine-specific plasticity occurring in response to glutamate uncaging. And basically, we have more receptors. It's actually, we show that it is actually more receptors on the spine, and volume is bigger, right? So this is what's happening. And because spine size is bigger, more receptor we have, more transmission occurs on this synapses, right? So this is a mechanism of synaptic plasticity. Now, how about intracellular signaling during this phenomenon? Just briefly explain what we found with the sensor before I'm going to explain about our new results, right? So basically, when we stimulate this single spine with the glutamate uncaging, we basically initiate calcium signaling, right? Activate a bunch of different proteins. And then actually, on the second stage, there some proteins activity spreads to surrounding spines. But this spreading signaling is not sufficient to induce plasticity in surrounding spine. But they kind of know, right? For example, this spine know that this spine is activated because of this spreading signal transaction. And some signals spread more, like PK spreads very long distance. And then sometimes it goes into all the way into the nucleus, activates the gene transcription. And of course, as I explained in the beginning, one of our motivations is the time scale of the signal transaction, right? So what we found is that when calcium elevated the spine, it's elevated in the spine, right? Calcium is very short life in the spine. They are removed, extruded from the spine almost immediately within 100 milliseconds. But then the signaling activate different proteins like Pkc or CANK2. So these are kind of calcium activated proteins. Or actually, calcium also induce release of peptides like BDNF. It's a peptide release that activates different proteins. And then it activates the second stage proteins like the GTPase proteins, CDC42, RAC, and RO. These proteins are activated, and their activation tends to be much longer. And then activate much later the nuclear activation. So we kind of understand what the time scale of the signal transaction. But today I'm going to talk about two different signal transactions. One is Pkc, the protein kinase C. This is a project of Leslie Colgan, who was a postdoc in my lab. So the protein kinase C, of course, probably many of you know the kinase. It's a very kind of old kinase we know of, right? Basically, it's activated by calcium, right? And it has kinase domain. But this calcium-sensitive Pkc is so-called classic Pkc. Actually, we have in the human genome we have 3 different isozymes: Pkc alpha, beta, and gamma. But actually, people found more different class of Pkc, right? Like novel Pkc, although it's a C probably means calcium. Novel Pkc is not calcium sensitive. It's actually sensitive to DAG. So classic Pkc is activated by calcium and DAG. Novel Pkc is activated only by DAG. Atypical PKC have all different regulation domain, right? Some of the particular kinase, like PKM data, which has only kinase site, there's no regulation domain, meaning that this kinase is regulated only by the translation or protein synthesis. So, PKC attracted a lot of attention in neuroscience because it's calcium sensitive, and in neurons, of course, everything is regulated by calcium. So people are excited that this enzyme might be important for learning memory. But then, because of this huge variation in the kinase family, actually, it has been pretty difficult to study how the kinase works. So it's a complex family of isozymes and existing two lacks. Isozyme specificity and spatial temporal discrimination was not possible. So let's decide to work on this. And let's see, basically developed sensors of PKCs, right? One is so-called IFRAC, isozyme translocation of C kinase. And another one is IDOX, isozyme specific docking of kinase substrate. So how it works and why it's substrate, it's isozyme specific. So basically, she fused the PKC isozyme with GFP. So this she used the whole enzyme here, just GFP fusion, and this GFP fusion does not impact the kinase activity or action of the kinase. So this GFP tag kinase is translocates the membrane when it's activated, and she also puts this membrane target RP. So when this binds to the membrane, they interact with each other and change the process lifetime due to FRET. Another mechanism, IDOX, is basically made of the pseudo substrate fused to RP and GFP fused to isozyme, one of isozyme of the kinase, right? When it's activated, they bind together and produce FRET. And then she went on to develop all different sensors for different isozymes. First series of studies, she worked on classic PKC, alpha, beta, and gamma. So this is image of process lifetime. Yeah? So basically, we measure process lifetime in each pixel and scans like this every 100 milliseconds, and then you can image the activity of the protein. This is a Pkc activation. You can see in real time, we uncage spines every one second. So you can see that actually in response to every uncaging, you observe the activation every 2 seconds, and activation of the Pkc. This is a pseudo color. So active Pkc gives us kind of red color. So you can see it's activating the spine. Pkc alpha activation is spine specific. So basically, this is a spatial profile. Yeah? So when engaged, there's some spreading to the dendrites, as you probably observed also in the movie. But actually, it doesn't go very beyond a micron or two. It's roughly a micron, actually. And it's also very fast kinase, right? Decays almost immediately. So it's activated for this is a second, right? 0.32 seconds, it's activated, and then it's already decayed around 1 second. This is a spatial profile. So activate spines, we have about 8% activation, and then decays to 0 within 1 micrometer. This is a temporal profile. Sorry that I forgot to put the axis. This is seconds. Right, so it's about 2 minutes of the time course. The vertical axis is activation, and the dots are uncaging. Yeah, so each uncaging provides us with activation of Pkc. isotype specific activation classic Pkc. So she measured alpha, beta, and gamma activation in the spine, and there's a plot of active Pkc. So you can see that both all alpha, beta, and gamma are activated on the spine and decays almost immediately within about 0.5 seconds. Among three of them, actually, Pkc alpha has strongest activation, and actually in this context, Pkc alpha is only important molecule for synaptic plasticity. Oh, is there a question?
Yes, Yasmin?
Yes.
Yes, I have a question. In this case, is the same, for example, the activation of Pkc in spines close to the proximal, close to the soma, or very distal ones?
Yeah, we measured there's different spines, and it's not dependent on the distance from the spine.
I see.
Or from the soma.
Okay, I see. And spine size, there is a correlation?
There is no correlation with our spine size.
Good.
Yeah.
Thank you.
Okay, Pkc alpha activation. Also, we can measure with substrate docking, and you can see actually it's slightly slower. The qualitatively, the activation pattern is very similar. Yeah? So it's activated, inactivated, activated, inactivated. Activation time profile is slightly slower because it's a different biochemical step, right? So they translocate to the membrane and then activate it. So, so we think that actually they are it's binding actually slightly slower. Okay, once you get the activation sensor, we can also measure what is upstream molecule. Yeah. So while we are measuring Pkc, we basically use a bunch of different inhibitors to probe what is upstream molecules. So in this case, we absolutely require glutamate receptor, which so-called NMDA type glutamate receptors that is calcium permeable. So when calcium comes into the spine, of course, the signal is initiated. So when we inhibit glutamate receptor, NMDA type glutamate receptors, we do not have any activation. This activation profile, and you can see that AP5, which blocks this NMDA receptor, we don't have any activation of this. Now, as I explained, Pkc alpha activation requires both calcium and DAG, and we wondered about what's the DAG source. And DAG is made of PIP2, which is cleaved by PLC. So we basically blocked PLC first with two different drugs. Both of them inhibited the Pkc activation. So PLC is required, which is not surprising. What did activated PLC? We found that actually one receptor called FACB, which is BDNF receptor, is required for Pkc activation. Now, 2016, we published a paper suggesting actually that BDNF is a peptide neurotrophic factor can be released from the spine and activate TrkB on the same spine. So we inhibited this signal transduction. So these two last two bars, 1 and PP1 is it's basically the method we can specifically inhibit the mutant mouse. I'm going to explain about this technique later, but basically just believe me that right now this technique allows us to just inhibit TrkB receptor only. And we found that this inhibits the signal transduction. Also, when we knock out BDNF from the spine, we also eliminate the signal transduction. So we are very sure that this autoclean signal starting from BDNF release from the spine and activating TrkB is required for synaptic plasticity. So, so what Leslie found in this paper is that actually Pkc integrates two different spatial temporal scale, right? One signal is TrkB, which provides DAG, deacylglycerol, right? And so TrkB activation, we show that 2016, when activates track B, actually lasting very long time, like 20 minutes. So it provides kind of long-term atmosphere in the dendrite. Actually, also there's some spreading to the nearby spines. Calcium is very fast signal, and right. So this is also we published a long time ago that calcium actually is it's very fast and decays within 100 milliseconds. It's much faster than the Pkc. A combination of these produce the Pkc activation. So it's basically a single molecule provide this computation in the spine. So it seems like actually we have a signal transduction occurring from calcium, which activates Pkc in millisecond time scale. Actually, there's a feedback from track B BDNF release, right, which activates track B receptor, activate PLC and so on forth, which is also required to be Pkc alpha activation. So there's a loop here, which is kind of interesting. What about novel Pkc? So Leslie also studied novel Pkc, which is activated only by BAG, not by calcium. Yeah. So first, she screened which novel Pkc is required for synaptic plasticity. So she used 3 different knockouts: epsilon knockout, eta knockout, and delta knockout, and measure the structural plasticity. Right. So this is a spine volume change in response to glutamate uncaging. Black is wild type, blue is not knockout. You can see that there's no change, especially we focus on this long-term structure plasticity. So measured between 20 to 30 minutes. Epsilon knockout, we didn't observe any difference. Eta knockout, we didn't observe any difference. And delta knockout, we observed the phenotype actually almost completely inhibited the structure plasticity, a long-term structure plasticity, which can be rescued by overexpressing Pkc delta only in postsynaptic cells. Okay. Now, we have also a technology to inhibit one specific kinase, right? So kinase inhibitor usually is not so specific, but if you can combine with a mutation onto Pkc ATP binding pocket, right, to make it slightly bigger. And this point mutation actually accepts a small molecule inhibitor called 1-NPP1. It's actually analog of ATP. It's very similar to ATP, but when this 1-NPP1 is provided, only that this mutation, right, analog sensitive mutation Pkc delta is inhibited, and all other ATP binding protein does not bind 1-NPP1. So it's actually selectively inhibit the mutated Pkc. So on the Pkc delta knockout background, we can overexpress this AS analog sensitive Pkc delta, and then we can inhibit basically inhibit Pkc delta acutely. So the blue is inhibition of Pkc delta from the beginning. Yeah? So you can see black is control. Gray is controlled with vehicle. The blue is 1-NPP1, and you can see that it is inhibited. When we apply 1-NPP1 after the induction, we didn't observe any inhibition. So it seems like Pkc delta is required between the induction and 10 minutes. So it's required for initial initiating the process, but not for the maintenance. Okay, so what the activation profile look like? So this is the activation of Pkc delta, right? She made sensors for all the Pkcs. You can see that it is activated in the spine. And it also slightly activates in the dendrite. It's actually not so clear by eyes. If we measure the activation, it looks like this. So the grades in the spine, you can see the activation and decay. In the dendrite, it also activated. Dendrite means that this whole dendrite is measured. And then actually we have kind of sustained activation. And I'm going to explain later what it does. Okay. Yeah. Okay. So now, because we know that we have the sensor, we also looked at the upstream molecule, and maybe you already kind of have predicted what happens, right? That this kinase is activated only by DAG, right? Pkc delta is activated by DAG. So just we need to track what produces DAG, and we already know that BNF track B system can provide DAG, and indeed, when we inhibit the signal transduction by track B, actually we inhibited the activation of Pkc delta almost completely. So we wondered what's the role of this long-lasting spreading. So let's wonder actually this might be the communication. It might be required for communication between the spine and the nucleus. So previously, we reported that stimulating 3 to 7 spines in one neuron, okay, can activate the gene transcription. We have 1,000 spines on each neuron, but just few spines, activating few spines is enough to activate the gene transcription. So we measured with ARC and also CREB. You can see that when we stimulate 7 spines in this case, the nuclear ARC is activated for a very long time, and this actually is Pkc dependent. So when we engage on spines, right, 7 spines, it takes some time. The nuclear ARC is activated, and then it's reversed by inhibiting Pkc. But this is non-specific Pkc, so we wondered actually Pkc delta is responsible for that. So let's measure the activation of Pkc delta on the primary dendrite, right? Stimulate 1 spine, that doesn't happen much. But when we stimulate 5 spines, we actually observe the activation in the primary dendrite. So yeah, so stimulating these 5, just to make sure that how we stimulate 1 spine, wait for 1 minute to settle, and go another spine and stimulate, and so on and so forth. So it's a very slow process. The neuron can accumulate such slow activation process. What happens downstream? Actually, we know that CREB, it's a transcription factor, CREB is activated, and we have the sensor for CREB. So we stimulate 5 spines and observe CREB activation. And then we inhibit Pkc delta here, right, after the induction, and we observe the very strong inhibition of CREB, CREB activation, suggesting that actually Pkc delta is required for activation of CREB in the nucleus. So it seems like Pkc delta has kind of 2 different roles. One is local LTP signal transduction, right? Basically, spine enlargement requires Pkc delta. But at the same time, it actually activates gene transcription, and probably important for extremely long-term signal transduction. Okay. Now, so this is Pkc delta. Now we know that Pkc delta is activated by just track B, and it basically translocates to the primary dendrites and activate the nucleus, gene transcription. Okay, the secondary story I'm going to talk about is insulin-like growth factor, which is a study of our grad student Mori too. She just graduated and going to different university, Berkeley, as a postdoc. So insulin-like growth factor, it's similar to insulin, right? But there are 3 peptides, very similar to insulin. One is insulin, and then IGF-1, IGF-2. And these 3 peptides are very similar. Acts as growth hormone, right? And IGF-1 and 2 activate IGF-1 receptor. Insulin actually activates insulin receptor. Yeah. So we wonder about this IGF-1 and 2 specifically. IGF-1 and 2 are synthesized in the liver, and some people found that actually some brain cells also can synthesize. IDX212 signaling is implicated in a lot of different diseases, diabetes, of course, but Alzheimer's disease, aging-related cognitive decline, and cognitive effects of exercise. So, so there are a lot of effects on our cognitive ability, right? So we wonder actually what's the action of IGF-1. IGF-1 signaling is basically transmitted by the peptide, insulin-like peptide, transmitted by insulin-like receptor, right? It's phosphorylates and recruit downstream molecule like Pkc kinase and important for metabolism, gene transcription, and plasticity. And there are a lot of autophosphorylation side, of course, but then when people knock out the PK signaling, it seems like it causes some problem in the cognitive capability. So we are interested in hippocampal IGF-1. So we wondered actually about IGF-1 signaling peptides localization first, where they are. And somebody found that actually IGF-1 seems to be mostly in CA1. So this is like hippocampus, CA1, CS3, and dentate gyrus. And you can see that expression is extremely strong only in CA1 regions. So we wondered actually, so she wondered if IGF-1 is necessary, and also at the same time we need to verify that this staining is specific, right? So we use the Frogs knockout, conditional knockout of IGF-1. IGF-1 knockout is with lethal, so we can't use just straight knockout. And on the conditional knockout, we removed sparsely using AAV flex GFP, right? It's conditional expression of GFP and CRE, right? So green cells have CRE, and red signal is from IGF-1. So you can see actually that we can actually label some of the neurons with CRE, and in these neurons, IGF-1 gene is knocked out. So when we go into this signal, green cells and how the staining look like, right? We found that actually the staining in these green cells are much reduced compared to for example control, right? For surrounding control first of all, but also in GFP. When we just infect with GFP, we didn't observe any reduction in the signal, right? So we think that IGF-1 actually is synthesized in these neurons and accumulates IGF-1 insulin-like growth factor. Let's skip this. And Mori also wonder if this the synthesis of IGF-1 is required for synaptic plasticity in the CA1 pyramidal neurons. So on the slides, she basically used again Frogs animal, conditional knockout, and using gene and sparsely transfect neurons. So only the postsynaptic cells, from the postsynaptic cell, we remove IGF-1 genes, but all the surrounding cells have IGF-1. So in this condition, right, the neuron we are observing does not have IGF-1 gene, and they can't synthesize it. And in that condition, we observe the attenuated plasticity. The black is control, green minus, red is positive, meaning that IGF-1 gene is removed, and we observed the phenotype, right, a reduced plasticity in IGF-1 knockout cell. And we could fully rescue this phenotype by infusing IGF-1 to the solution. So it suggests that IGF-1 release from or IGF-1 synthesis in this neuron is necessary for synaptic plasticity. Mori teamed up with Anna Jane, our postdoc, to measure also the glutamic glutamate sensitivity by glutamate receptor plasticity, or we call it actually electrophysiological LTP, long-term potentiation. And basically, knockouts, the control cells, we can induce LTP. So basically, what we observe is an increase of synaptic current in response to stimulation. Yeah. When we remove 3, we didn't observe potentiation of the current. So this suggests that this type of synaptic plasticity requires IGF-1 gene. At this point, basically, Mori decided to make sensor of IGF-1 receptor. So she put GFP to the IGF-1 receptor, and downstream molecule PI3 kinase, actually the fragment of PI3 kinase with RFP, SH2 domain actually. So when phosphorylation occurs, this SH2 domain is recruited to the phosphorylated site and therefore produces spread. And by using this, we can measure the IGF-1 concentration in the cell, on the surface of the cell. So in the neuron, we transfected with this sensor and are fused with IGF-1. And you can see that sensor gives us IGF-1 concentration. Right? So at 6 nanomolar, we are pretty much saturating IGF-1 signal. So basically, we observe activation of IGF-1 receptor in dose response manner with IGF-1 concentration. So using this sensor, we observe the activation of IGF-1 in response to single spine anchoring. And basically, to our surprise, it's activated on the spine that is stimulated, suggesting that IGF-1 is probably released from the spine and activating IGF-1 receptor on the spine. And also there is spreading, and IGF-1 release the IGF-1 might spread actually over several microns and activate surrounding spine. When we inhibit glutamate receptor, we didn't observe any activation of IGF-1 receptor, suggesting that calcium through glutamate receptor is responsible for IGF-1 receptor activation. We also removed the IGF-1 gene from the postsynaptic cell to make sure that actually IGF-1 synthesis is required for activation of IGF-1 receptor on the spine. And indeed, when we removed IGF-1 gene from the neuron, we observed the reduction of the activation of IGF-1. So finally, Mori basically decided to measure IGF-1 release more directly by using slice culture of hippocampal neurons and measure the IGF-1 release to the medium. And basically, in the slice incubated by sodium compared to potassium, right? We observed a larger release of IGF-1. So IGF-1 is released in activity-dependent manner to the medium. When we remove the IGF-1 gene from the pyramidal neurons using the CAM-K2 promoter, which this is excitatory neuron specific, so only removing IGF-1 gene from these neurons, we observe the reduction. So therefore, what we found is, what Mori found is that C1 pyramidal neurons are the major source of IGF-1, and IGF-1 is released from the spines in NMD-dependent manner. And local autocrine, right? So basically, calcium influx causes a release of IGF-1 from the spine, and this comes back to the receptor, and this signal transfer probably activating DTPase proteins, a bunch of DTPase proteins that activate the cytoskeletal regulation. So, basically, we found actually new signal transduction, right? Surprisingly, through insulin-like growth factor. Insulin-like growth factor also is circulating and comes in and get into the brain through blood-brain barrier. It seems like actually each dendritic spine also provide local IGF-1 to probably quickly activating the activating the IGF-1 receptor and coding synaptic rapid synaptic plasticity. So with that, want to finish my talk by showing this diagram of protein activation. We measured a bunch of different proteins, how they are activated, and measured the time course. And you can see that actually how they are how they work, right? From the calcium, so we start from calcium here, this calcium signal, and also BDNF release occurs here, and activating the BDNF surrounding proteins. At the same time, activating part of a signal transduction and also acting regulation proteins. There are some activation in the nucleus, of course, and endosome. Activation is required for exocytosis of a bunch of different proteins, including glutamate receptors and membrane. So basically, we start to understand this complex signal transduction system in the spine, and next goal is to basically understand how this is related to actual animals learning and memory. With that, I finish my talk. I especially thank who contributed to this the particular work led by Leslie Colgan and Mori Tu and Anna Jane. These three people particularly contributed to these works. And thank you for your attention.
Thank you so much, Ryohei. It was a wonderful talk. It's amazing how many tools you have developed and the results you have gotten with them. Now I would like to open the floor for questions. So if anyone in the public has any questions, please. I think they are shy.
I have a question if someone.
I would check YouTube to see if there is any questions there. I think not yet. Yeah, please, Jasmine.
Thank you so much. Thank you, Dr. Yasuda, for the amazing talk. I have many questions, but I'm going to start for one. In the activation of PKC, when you activate some different spines, for the, the induction of the transcription, seems that dendritic domains are not then particularly important for trigger that response. Is that right? Because you stimulate in different dendrites, right? As far as I understood.
Sorry, I didn't. Can you elaborate your question? So
sorry.
It's initiated in the spine, and that spreads into the dendrite, and that actually gets into the primary dendrite, activating gene transcription. So, and gene transcription probably will have significant impact eventually to the synaptic plasticity. But this quick synaptic plasticity, it does not have actually any role.
Okay.
It will be important for very extremely long-term signal, which we cannot measure in this system.
I see. But what I mean is that, for example, if the activation of the spines instant, you do like in a spread spines, you choose a dendritic domain, then the response could be a long-lasting response.
Dendritic domain. You mean activating dendrites directly?
No, dendrites, spines.
Yeah.
In a region of a specific region of a dendrite. You know what I mean?
I think I understood. So you are asking if we stimulate, for example, just from one branch? Yeah. So we have done the experiments. So this paradigm requires to activate multiple dendrites. It is very interesting. So if we stimulate all 5 spines on the same dendrite, then this cannot occur. We need to distribute to different branches. And then the transcription occurs. So it's a very interesting system actually that it basically selectively enhance the stimulation that is distributed spatially.
Yeah, I see. And can I ask another one? Okay. So the activation and all the molecular pathways that you are seeing now with these amazing tools, have you proved this in the LTD protocol, with an LTD protocol?
It is a very interesting question, and we are developing a LTD protocol right now, but we haven't done yet.
Oh,
It is an important question, so
yeah,
hope that we can do it soon. It is not easy in Florida. Just everybody's too happy. So
yeah, well, maybe soon you have protocols from Invali Israeli lab coming, hopefully. And I have another one.
Yeah.
For if you do, do you see difference, for example, if you instant to do the regular LTP protocol with high stimulation, engaging high stimulation, if you do like STDP protocol, do you see the same? Or have you also tried to test that?
Yeah, I want to test that too. We don't know it. We don't have the answer yet. Yeah. Well, to do STDP, yeah. Well, we need to do that, but just that we haven't done that yet.
Yeah, that will be really nice.
Yeah.
Okay. Thank you so much.
Thank you for the question.
Thank you, Jess, and thank you, Ryohei. So Jasmine is our dendritic spine specialist in the institute.
Oh, good.
Sorry, I think my internet is cutting a little bit, but I don't know if there is any other question. I don't know, Ryohei, if you have been exploring the behavioral impact of the modification of these proteins. Mm-hmm.
Yeah. That's also a very important question. We are working on it, especially for IDF1. It is very specific to CA1 region, and we hope that the phenotype must be very specific to CA1. So we are knocking out from pyramidal neurons. Unfortunately, we knock out from all pyramidal neurons, but probably the effect should be only CA1. So we are just checking the behavioral impacts right now. So far, it seems like it's like very mild actually, because it's only part of the brain. So the impact is not so strong right now.
I see, but I think it would be very interesting to see in some pathological conditions, right?
Yeah, probably
this could be exacerbated or.
Yeah, that's probably will be more informative, I think. Actually, I'm pretty sure that there's some effects if we combine with some disease model, for example. But yeah, right now, yeah, we are not. We are working on behavioral aspects.
That's great. I think that will be an amazing talk to listen in a few years, right?
Hopefully so.
We can invite you again.
Yeah.
Yeah, so I think if there are no more questions, well, we thank you very much for accepting the invitation, Ryohei, and we hope to have you here in Mexico soon. And thank you very, very much for giving this amazing talk.
Thank you very much.
Thank you.
Thank you very much.
See you.
Thank you.
Can you finish the meeting for me? It seems like I can't.
Yeah, we will. Yvette, if you can just